Hello, welcome to my YouTube channel. Today we will learn about type 4 hypersensitivity reaction. Type 4 hypersensitivity reaction is mediated by T lymphocyte including CD4 and CD8 T cells. It is an example of delayed type 48 to 72 hours after exposure of antigen takes place. Type of type 4 hypersensitivity reaction is cytokine mediated inflammation. CD4 T cell produce cytokine in this condition and cause inflammation. Direct cell cytotoxicity, it is mediated by CD8 T cells. Mechanism: intracellular bacteria or antigen presented by antigen presenting cell to CD4 cells. CD4 cell get differentiated into delayed type hypersensitivity T cells, which generally is Th1 cell. Sometimes it may be Th17, Th2 and CD8 cell occasionally. Here is this is here is sensitization phase is completed when second dose of the same antigen get entered in the body or encountered with Th1 cells. Th1 cell secrete interferon gamma and membrane TNF beta, and it activates resting macrophages. Resting macrophage get activated and it have class second MSC receptor, TNF receptor. Activated macrophage cause phagocytosis of the antigen. Effect of macrophage activation is increased class second MSC molecule, increased TNF receptor, increased oxygen radical and increased nitric oxide. These components enhance the phagocytic activity of macrophages or other phagocytic cells. Delayed type hypersensitivity cells secrete cytokine interferon gamma, which is chief cytokine, interleukin 2, TNF beta, MCAF, MIF, interleukin 3, and GM-CSF. Now we will learn about these cytokine which secreted by delayed type hypersensitivity T cell in detail. What is their function? Now we will see cytokine secreted from a delayed type hypersensitivity T cell. Interferon gamma, it is a key cytokine of type 4 hypersensitivity reaction. Activate the resting macrophage into active macrophage. Active macrophage highly competent to microbial killing by various mechanisms such as increased expression of MSC, second, increased TNF receptor, increased oxygen radicals and nitric oxide. Interleukin 2 act as an autocrine. Autocrine stimulate the proliferation of delayed type hypersensitive cell. Autocrine is this chemical interleukin secreted by delayed type hypersensitivity T cells, and it act on the same cell, delayed type hypersensitive cell, and stimulate their proliferation. MCAF, monocyte chemotactic and activating factor. It along with TNF beta help in migration of monocyte from blood to site of delayed type hypersensitivity. Transform into macrophages. MIF, migration inhibitory factor, inhibit migration of macrophage from the site of delayed type hypersensitivity, which means macrophage, this cytokine prevent the migration of macrophage from the site, prevent escape of macrophage from the site of delayed type hypersensitivity reaction. Interleukin 3 and GM-CSF. GM-CSF means granulocyte monocyte colony stimulating factor. It helps in local synthesis of monocytes. Now we will see in detail about type 4 hypersensitivity reaction. So first is cytokine or cell-mediated inflammation. Cytokine produced by the CD4 T cell is responsible for CD4 T cell mediated hypersensitivity reaction. Cytokine can induce chronic and destructive inflammation. Naive or native CD4 cell recognize antigen displayed by antigen presenting cells and secrete interleukin 2, which act as autocrine growth factor. It stimulates the growth of this CD4 T cells. CD4 cells get differentiation of as naive cell into effector CD4 T cell. Activated CD4 cell produce activated CD4 cell get differentiated into Th1 cell and Th17 cell. Here Th1 cell is promoted by interleukin 12, which secreted by antigen presenting cell, and here CD4 effector CD4 T cell get differentiated into Th17 cell when it induced by interleukin 1 and interleukin 6 and interleukin 23, which secreted by antigen presenting cells. Now we will see this. Th1 cells secrete cytokine interferon gamma and TNF, which activate macrophage. Activation of macrophage and inflammation leads to destruction of intracellular microbe and tissue damage. This response takes place against intracellular pathogens like viruses and some bacteria. Promote inflammation and activating other immune mechanism. Th17 cell secrete interleukin 17, interleukin 22 and other chemokines. It helps in recruitment of neutrophil at the site of delayed type of hypersensitivity. It promotes inflammation. This response is produced against extracellular bacteria and fungi. Example: contact dermatitis and tuberculin test. This is a type of cell-mediated inflammation. When this antigen or foreign body is persistent or non-biodegradable. Example: in case of mycobacterial tuberculosis, naive CD4 T cell recognize antigen displayed by antigen presenting cell. Differentiate and proliferation of naive CD4 T cell into CD4 plus Th1 cell. Then CD4 Th1 cell activation leads to activation of macrophage. Transformation of macrophage into epithelial cells. Then granuloma formation is seen. Granuloma Granuloma is a microscopic aggregate of epithelioid cell surrounded by a rim of lymphocyte. These are outer layer is fibroblast, inner is a lymphocyte, and here is epithelioid cell. This large cell is Langerhans giant cell. And this part is gaseous necrosis from this picture. Now we will see second type of type 4 hypersensitivity reaction. Here, antigen bearing target cell. This response is this type of hypersensitivity type 4 occur in case of antigen bearing target cell or surface mounted antigen. Example, virus infected cell and tumor cells. Here, then CD8 T cell recognize antigen displayed by antigen presenting cell. CD8 T cell, also called as cytotoxic T lymphocyte, which in short we had written is CTL, get activated. CTL or cytotoxic T lymphocyte releases perforin and grain enzyme. Perforin produce transmembrane pore forming molecule, allow entry of grain enzyme into target cell cytoplasm. Grain enzyme, it is a protease which cleave and activate cellular caspase. Caspase promote the apoptosis of target cell through fast ligand activated cytotoxic T lymphocyte, which have fast ligand, plus target cell which have fast receptor CD95. These react come in contact promote or start the process of apoptosis. There are two pathway of apoptosis, extrinsic and intrinsic pathway. Here is extrinsic pathway of apoptosis get activated. Example of type 4 hypersensitivity reaction is rheumatoid arthritis. Here, antigen is collagen with a collagen fiber, and this is cytokinated cell protein act as antigen. Multiple sclerosis, where myelin derived protein like myelin bound protein, phospholipid, phospholipoprotein, and myelin glycoprotein, and type 1 diabetes, antigen of pancreatic and beta cell of isolate. Inflammatory bowel disease, here enterobacteria act as a self-antigen. Peripheral neuropathy, here myelin associated protein is antigen. Hashimoto thyroiditis, thyroglobulin and other thyroid protein act as antigen. Crohn's disease, microbial antigen like flagellin and autoantigen like GP2 act as antigen. Other example is graft versus host disease, lepra reaction type 1, intracellular bacteria or fungi or virus. In case of intracellular bacteria or fungi or virus, type 4 hypersensitivity reaction occurs. In granuloma formation also seen this type 4 hypersensitivity reaction. This is continuation with cytokine mediated type 4 hypersensitivity. Here, after what happened after inflammation get over in type 4 hypersensitivity reaction, as we see in cytokine or cell mediated inflammation. After inflammation get cleared, the effector T cell convert into memory T cell, which is long lived respond quickly if the same antigen is encountered again. Th1 cell produce interferon. Upon repeated exposure to antigen, Th1 cell produce interferon gamma. Interferon activated macrophage, classically activated macrophage, produce four responses: increase ability to phagocytose and kill, express more class 2 MHC on surface, increase antigen presentation, secrete TNF and interleukin 1 and chemokine, promote inflammation, and this promote more interleukin 12 secretion. Interleukin 12 secretion further amplified this Th1 cell proliferation. Thanks for watching. Subscribe my YouTube channel for further upcoming videos. Thanks.